So this is the very, very basics, and again, I hope that today this will be a review. This little first section is just something I wanted to comment on. It's not actually in the notes, but remember that genetics is based on probability. When you make a Punnett square and you predict that you expect 3/4 of the offspring to come out tall and 1/4 to come out short, that doesn't mean that if there's 4 offspring, it definitely has to be 3 tall and 1 short. And the same thing like when you flip a coin. Just because there's a 1 out of 2 chance doesn't mean that if you flip a coin twice, you're definitely going to get 1 heads and 1 tails. The fact of the matter is, the more flips you do, in general, statistically, you will come out to approximately 50-50. If you do lots and lots of flips, typically you will end up pretty close. But if you only flip it 10 times, you may or may not come close to that 50-50 prediction. So you should know how to figure out a prediction or how to get the probability for something happening. And we already talked about coins. You did this yesterday. The reason why the chance of flipping heads is 1 out of 2 is because how many sides does the coin have? 2, and only one of those sides is heads. So that's where the chance is 1 out of 2. What about the chance of rolling 6 on a die? Right, somebody said 1 out of 6. Because there's 6 sides on the die, and only one of those sides has a 6. Now what if I said, what are the chances of rolling either a 6 or a 5? Now what are your chances? 2 out of 6. Because it still has 6 sides, and they're saying that there's 2 things you could get. You could get a 6 or you could get a 5, and in that case, both of the, either one of those would satisfy the requirement. Now here's where it gets a little tricky, and I'm going to show you a rule to answer this question if you don't already know the answer. I asked this earlier today, what are the chances of rolling a 6 on 2 dice? In other words, you roll 2 dice and they both come out 6. And I've had a couple of different answers throughout the day. Some people told me 2 out of 6, because they figured now you've rolled it twice. I had a couple of people that said 1 out of 12, because they added the denominators together. Don't ever do that. Don't ever, ever, ever do that. And some people thought 1 out of 36, you multiply the denominators together. So these are the 3 possible answers that I got, and one of them is correct. So the rule is that I want you to use, and this rule is actually on the formula sheet that you can use on the AP exam. The rule is called the product rule, and it basically says that the chance of any 2 events both happening are their individual chances multiplied together. So the correct answer to that other question is 1 out of 36. So you roll up the first die, and you get a 1 out of 6 chance of getting a 6. You roll a second die, you also have a 1 out of 6 chance of getting a 6. So the chance that when you roll both dice, you get a 6, it actually is 1 out of 36. Now, just to see that you understand this, and then we're going to apply it to genetics later, it says here, what are the chances of rolling a die and flipping a coin at the same time, getting a 5 on the die, and getting heads on the coin? So we do the same thing, product rule. What are the chances of getting a 5 when you roll a die? Still 1 out of 6. And what are the chances of getting heads on the coin? 1 out of 2. Therefore, what are the chances of both of those things happening? 1 out of 12. So we can apply this rule to genetics. It's pretty easy to use, and it will make some problems in genetics that, for example, if you tried to do a Punnett square, would be very tedious. This will make those problems really, really easy to answer. So it's called the product rule. Chance of any 2 things both happening would be their individual chances multiplied together. All right, so that brings us to our review stuff. So Mendel is considered the father of genetics. Anybody remember what organism he studied?
Plants.
Yes, he studied pea plants specifically. He was a monk, and he is the father of genetics because he was basically the first person to really do experiments in genetics, in inheritance. And he did work with garden peas. Now it turns out that the fact that he worked with garden peas is really important, because had he been looking at other organisms, he might not have been able to make the discoveries he made. Because a lot of organisms are very complicated to study. If you knew absolutely nothing about genes or chromosomes or anything else, and you tried to study people, for example, it would be very hard to figure out how we passed on certain traits, like hair color. You'd have 2 people that are light brown hair, and then they have a red-headed child, and it's like, how in the world do we explain that? It's not a simple explanation. The same thing for our heights. It's not like if you just cross tall and short, you get medium. Not necessarily. There's this whole range of heights where you could have 3 offspring in a family and one's 4 inches taller than the other one, and it's just much more complicated. But pea plants are really easy to study. And there were a couple of reasons why pea plants worked out really, really well. First of all, pea plants are self-pollinating. What that means is, it says you can assure the paternity, meaning who the father is. You could purposely cross pea plants with each other. But here's what it means to be self-pollinating. In other words, this is the female part of a flower, and then the pea plants happen to be hermaphrodites. They also had male parts on the same flower. So it looks something like this. So what you could actually do in a pea plant is you could take pollen from this plant and fertilize the same plant. And that's called self-pollination. Now you might say, well, how is that sexual reproduction? Isn't that asexual? It's the same plant. But technically it's not. It is technically the same plant, but it's not asexual reproduction because you still have sperm, because that's what pollen is. You have sperm and you have eggs, and you have sperm fertilizing an egg. And let's imagine this. Let's say that this plant was big B, little b for a particular trait. Wouldn't it be true then that when the egg is made, it can either give a big B or a little b? And when the sperm is made, it can also give either a big B or a little b. And that means that it would be possible that this plant, that's big B, little b for a particular trait, could have an offspring that was little b. So technically this is sexual reproduction. You don't see it in animals much. There are a few animals that are hermaphrodites, like sponges, I think some flatworms, and things like that. But in plants, you do see this, and this would be considered sexual reproduction. And so he could easily control the mating in the plants. He could just let it fertilize itself, and that way he would know that he was crossing this with the same thing. Or if he wanted to cross it with another breed, he could just cut off the male parts on this plant. Sounds kind of harsh, but in plants, I don't think they care. And then he could take the male parts from another plant and then just purposely take the pollen and then fertilize this plant. So he had a lot of control over the crossing of the parents. Secondly, the traits were really easy to see. Like I just said a minute ago, human traits are complicated. You can't necessarily look at every human trait and see a very simple, oh, you either have it or you don't. There are some cases like that, but with pea plants, every trait he looked at worked the same way. The flowers only came in 2 colors. They were either purple or white. You didn't get shades in between, nothing like that. The plants were either tall or short. There was no range of heights like what we have. It was either they were tall or they were short. The seeds were either wrinkled or they were smooth. So it was really easy to look at the traits in the pea plants, and that allowed him to get a better idea of what was going on when the traits were being passed on. And finally, and this is probably the most important thing, they grew really fast. They were very easy to grow. They didn't require a lot of maintenance, and they made lots and lots of offspring at once. Because if you think of a pea plant, its offspring are the peas that it makes. Which means if that pea plant makes 10 pods and every pod has 10 peas in it, there's 100 offspring right there that you could then plant and get the next generation going. On the AP exam one year, there was a question like this, where they gave a cross and they explained that they took 50 individuals of one and 50 individuals of another, and they crossed them together and ended up with like 200 offspring, and they crossed those and went through this whole thing. And the very first question that it asked was, what organism could have been used for this experiment? Now, pea plants would have been an appropriate answer. That would have been fine. But can you think of an animal based on this that would also fit the criteria? Go ahead. Bunnies would be perfect. Mice, rats, flies, anything that would make lots of babies, would be fairly easy to take care of, and they grow fast. So they were looking that you would come up with some kind of organism that made lots of offspring, that reproduced sexually, made lots of offspring. Humans would be a very poor example. We only have a couple offspring. We're not easy to take care of. We do not grow fast. Our traits are not easy to see. And not only that, but you can't necessarily control the matings and go, hey, let's mate you with you and let's see what we get. People aren't going to do that. So it's much more difficult to study humans. The fact that he studied pea plants is really, really important to the discoveries that he made. All right, so what did he do? Basically, this is what he did. He would take 2 pea plants that were purebred, meaning they always made offspring that looked the same. and he crossed them with each other to see what would happen. So we're going to use the sample of a round seeded plant crossed with a wrinkled seed plant. So what would happen is, in this particular cross, when he crossed round and wrinkled, he found that all the offspring of this cross were plants that made round seeds. At the time, they believed in what was called the blending of stuff. In other words, if you crossed round and wrinkled and round came out, it was because round and wrinkled mixed together, and round was just so strong that wrinkled was wiped out, and wrinkled would never, ever, 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 ever appear again. They would just be round from then on, because round sort of just wiped it out. But Mendel showed that wasn't what was happening, because what he then did was he crossed two of these with each other. In this case, with plants, remember, he could let them self-pollinate, meaning that would basically allow two of them to cross with each other. And when he did this, the next generation came out approximately 3/4 round and 1/4 wrinkled. So he was able to show that this trait of wrinkled, it didn't disappear. It was sort of hidden in the second generation, but then it could come back, and you could see it come back again. So there wasn't a blending or a disappearing of the trait for wrinkled. So he came up with some vocabulary that went along with this. For one thing, what did he call it, or what do we call it today when one trait covers up another one? Dominant. So he basically is the one that came up with the words dominant and recessive. He said, okay, so the trait that actually covers the other one up, he would call that dominant, and the one that got covered up, he would call recessive. So he was the one that came up with those vocabulary words. Before we go into, he came up with 3 principles from this, but before we do that, I have this rewritten, and I have one other thing I want you to know about it. So this is rewritten what we already did. Round times wrinkled, they all came out round, and then you got like a 3/4, 1/4 ratio in the next generation. The vocabulary I want you to know from this, or add to this, would be these 3 things though. P, F1, and F2. When he did his crosses, and even today, when scientists do genetic crosses, if they start with 2 purebreds, they always call that the P generation. It stands for parents. So this is the original parents, the parental generation. This only applies when a scientist is starting fresh with purebreds and then doing the very first cross. Their offspring are called the F1 generation. It would be nice if they were like the O generation for offspring, or C for children or something. But remember, these studies were done in the 1800s, and he was a monk, and so his vocabulary was coming from Latin roots. And filial means son or child. So the first filial generation, the F1, is the children of the original parents. And then the next generation, he just called it F2. The second filial generation, meaning let's carry this on and let these offspring go to the next generation. Sometimes you actually see it written like this, with the 1 and the 2 as subscripts, just so you know that's okay too. So that's the only other vocabulary I wanted to add to this slide. So that's basically what he did in his crosses. He would cross 2 things, and then one of them would always cover up the other one in every single trait he tested. And then in the next generation, he would get this 75%, 25% ratio. And like I said, if he hadn't done pea plants, he might not have been able to make these discoveries. Pea plants were just very simple. So when he crossed purple flowers and white flowers, they all came out purple. Then he got 3/4 to 1/4 after that. When he crossed green seeds and yellow seeds, they all came out green. Then he got 3/4 to 1/4 after that. So consistently, every trait he looked at, he was able to see what he called the principle of dominance. And that was his first principle, the principle of dominance, which basically just says that some traits, he called them characters, masked others. The ones that did the masking were called dominant, and the ones that got covered up were called recessive. He didn't know about chromosomes. He never used the word genes. He didn't know about meiosis. Today, though, we do. So today we would say that this is a gene for a trait, and then we would call the dominant form and the recessive form alleles. Oh, in fact, I think I actually have it on the next line here. So our vocabulary today, we would say that there was a gene for seed shape, and I actually had used round before instead of smooth. And we would say that it had two alleles. There was one allele that coded for round and one allele that coded for wrinkle.